name is Emily Jas, and today the topic I am going to discuss with you all is genetic linkage. So before starting, I have a question that is independent assortment always the case? So the answer is no. No, the independent assortment is not always the case. According to the independent assortment, during gamete formation, the two alleles for one gene segregate or assort independently of the alleles of the other genes. But if two genes are found on the same chromosome, they will not assort independently and do not follow the Mendelian inheritance pattern. Genes that are inherited together are said to be linked. Genes located on different chromosomes are not linked. This shows or allows independent assortment, and such trait shows the classic 9:3:3:1 inheritance pattern. Genes that are located very close together on the same chromosome, they may show complete linkage. On the other hand, genes located far apart on the same chromosome typically show incomplete linkage. So now we will see the genetic linkage definition. Genetic linkage is the tendency of alleles that are located close together on the chromosome or very nearly present on a chromosome to be inherited together during meiosis. Here you can see in the diagram, genes whose loci are nearer to each other are less likely to be separated onto different chromatid during chromosomal crossover or meiosis. On the other hand, those genes which are less closely linked have more chances of separation or segregation during meiosis. So how the genetic linkage was discovered? Two scientists, William Bateson and Reginald Punnett, completed a study in 1905, and they determined the movement of alleles found on the same chromosome and that how these alleles present on the same chromosome moved. They especially studied the sweet peas and studied their two characters, particularly flower color and pollen shape. In sweet peas plant, we see the dominant trait are purple color and long pollen shape. On the other hand, the recessive trait are red color and round pollen shape. Now you can see that in parental generation, when two true breeding or homozygote strains are crossed after fertilization, we can see that in F1 generation, the purple flower and long pollen traits are dominant. Purple color is dominant over red, and long pollen is dominant over round pollen. So all the F1 generation was 100% purple long. On the other hand, when this F1 generation purple flower with long pollen is self-fertilized, we would in F2 generation we would observe four phenotypes. First phenotype is purple petals with long pollen, round pollen with purple petals, red petals with long pollens, and red petals with round pollen. And their phenotypic ratio is 284:21:21 to 55, which will not follow the common pattern of 9:3:3:1, and these alleles did not assort independently and therefore must be linked. So we can say that the alleles for flower color and pollen shape is present on the same chromosome, and these genes are linked. Another example was given by Thomas Hunt Morgan. One day in 1910, Thomas Hunt Morgan peered through a hand lens at a male fruit fly, and he discovered an abnormality. He discovered that instead of having normal brilliant red eyes, fruit fly has white eyes. So he examined two traits, body color and wing color. In fruit flies, dominant traits are gray body represented by B positive and normal wings represented by VG positive. On the other hand, the recessive traits are black body B and vestigial wings presented as VG. So in the parental generation or P1, when a true breeding grey body with normal wings is crossed with black body and vestigial wings, after fertilization, all the offspring in F1 generation are um, grey body and normal wings. The grey body and normal wings are dominant over black body and vestigial wings. When this gray body and normal wings is crossed with a homozygous recessive for a test cross, we have four phenotypes. um, Two phenotypes are normal offspring and two phenotypes are recombinant offsprings. In the results, we can say that in F1 generation we have four phenotypes: gray normal, black vestigial, gray vestigial, and black normal. And we expect that expected frequency would be 575 for each, but observed frequencies are 965 for gray normal, 944 for black vestigial, and 206 for gray vestigial, and 185 for black normal. So we can say that two are parental phenotypes and two are recombinant phenotypes. Or so as a result, we can say that the gene for body color and wing shape is present on the same chromosome. So we can say that um, the gene for body color and wing shape is present on the same chromosome, and sometimes um, um, sometimes these genes do not assort independently and become and show recombination during meiosis or crossover. So this will shows the genetic linkage that two genes are linked and these two linked genes are not separated during meiosis or chromosomal crossover. I hope you understand the concept. Thank you.